There is a moment happening right now that most people never think about. Your heart is beating, your lungs are expanding, and somewhere in your body, a muscle is contracting. Not because you commanded it, not because you thought about it, but because of a process so ancient and so precise that it has remained essentially unchanged for hundreds of millions of years. The question of how muscles actually move is not a simple one. And the answer, when you look closely, is far stranger and more elegant than anything you might expect. Most people grow up with a rough idea of how movement works. You think about moving your arm and your arm moves. Simple, intuitive. But that description skips over what is arguably one of the most sophisticated mechanical systems ever produced by biological evolution. Between the thought and the movement lies a cascade of events, electrical, chemical, and physical, that unfolds in fractions of a second and involves structures so small they were invisible to science until the 20th century. Understanding this process means understanding something fundamental, not just about the body, but about life itself. The human body contains more than 600 skeletal muscles. These are the muscles you consciously control. The ones that move your limbs, hold you upright, allow you to speak, swallow, and breathe. Together, they account for somewhere between 30 and 40% of your total body weight. But raw numbers do not capture the real picture. The real picture is one of extraordinary coordination. Every time you take a step, dozens of muscles activate in a precise sequence, some contracting, some relaxing, some stabilizing, all working together without any conscious orchestration on your part. The body does this automatically, fluidly, and almost always without error. For most of human history, the mechanism behind this was a complete mystery. Ancient physicians knew that muscles produced movement, but they had no way of knowing how. The Greek physician Galen, working in the 2nd century, believed that muscles were activated by a form of pneuma, a vital spirit that flowed through the nerves like liquid through a pipe. This idea, elegant in its simplicity, dominated medical thinking for over 1,000 years. It was wrong, but it was not entirely unreasonable for its time. The nerves do carry something to the muscles. It is just not a liquid. It is electricity. The understanding that nerves transmit electrical signals began to emerge seriously in the 18th century, with experiments that sent currents through severed frog legs and watched the muscles twitch in response. This was disturbing and fascinating in equal measure. It suggested that the life of a muscle, its capacity to move, was somehow electrical in nature. But electricity is a property of circuits and wires. How does a living cell use it? The answer begins at the level of a single nerve fiber. A motor neuron, the type of nerve cell that carries commands from the brain and spinal cord to the muscles, is a remarkable structure. It has a long, slender extension called an axon that can stretch several feet in the human body, traveling from the spinal cord all the way down to the muscles of the foot. Along this axon, the neuron conducts what is called an action potential, a wave of electrical activity that travels at speeds of up to roughly 300 feet per second, depending on the type of fiber. This is not electricity in the way a wire carries it. It is a biochemical event, a rapid shift in the distribution of charged particles across the cell membrane, traveling like a domino effect from one end of the neuron to the other. When this electrical wave reaches the end of the motor neuron, it arrives at a structure called the neuromuscular junction. This is where the nervous system hands off its message to the muscular system. And what happens here is one of the most studied and most important biological interfaces in the entire human body. The tip of the motor neuron does not physically touch the muscle fiber. Instead, there's a tiny gap between them, a synaptic cleft. When the electrical signal arrives, it triggers the release of a chemical messenger called acetylcholine. These molecules drift across the gap and bind to receptors on the surface of the muscle cell. When enough of them bind, they trigger a new electrical signal inside the muscle fiber itself. The command has been received. But here is where the story deepens. That electrical signal on the surface of the muscle fiber is just the beginning. The muscle cell has an elaborate internal architecture, and the surface signal needs to travel deep inside it to trigger what actually produces force. Running through the interior of each muscle fiber is a network of tubular structures called T-tubules, transverse tubules that carry the electrical signal from the surface membrane inward, down into the core of the cell. There, the signal reaches a structure called the sarcoplasmic reticulum, which functions as a kind of reservoir for calcium ions. When the electrical wave arrives, the sarcoplasmic reticulum releases a flood of calcium into the interior of the muscle fiber. This calcium is the trigger, and what it triggers is one of the most elegant molecular machines in biology. Inside every muscle fiber are thousands of thread-like structures called myofibrils. Each myofibril is composed of repeating units called sarcomeres, and the sarcoma is the fundamental contractile unit of muscle. Under an electron microscope, sarcomeres appear as alternating light and dark bands. A pattern so regular and precise, it looks almost artificial. These bands correspond to two different types of protein filaments arranged in a specific geometry. The thick filaments are made of a protein called myosin. The thin filaments are made primarily of a protein called actin. The central insight of muscle biology, the idea that changed everything, is that contraction occurs when these two sets of filaments slide past each other. This is known as the sliding filament theory, and it was proposed independently by two groups of scientists in 1954. Before this discovery, the dominant assumption was that muscle fibers must shorten by some kind of coiling or folding mechanism. The idea that the filaments themselves do not change in length but simply slide relative to one another was a genuine scientific revolution. It explained for the first time the precise mechanical basis of muscular force. But how does the sliding happen? This is where the story arrives at one of the most extraordinary molecular machines ever described. Each myosin molecule has a structure that resembles a golf club, a long tail and a rounded head. The myosin heads, which protrude from the thick filaments, are capable of reaching out, grabbing an actin filament, and pulling it. They do this through a motion called the power stroke. The myosin head binds to actin, bends at a hinge-like joint, pulls the actin filament along by a tiny increment, and then releases. It then resets, reaches forward again, and repeats. This cycle happens millions of times per second across the length of a single muscle fiber, with countless myosin heads working in a synchronous rhythm so that at least some are always pulling at any given moment, like an endless team of rowers pulling on oars. The energy for this process comes from ATP, adenosine triphosphate, the universal energy currency of the cell. Each pull of a myosin head requires one ATP molecule, which is broken down to release energy and then must be regenerated. This is why sustained muscular effort requires continuous metabolic support. The burning sensation of prolonged exertion is partly the consequence of the extraordinary metabolic demand that muscle contraction places on the cell's energy systems. Now consider what calcium has to do with all of this. In a resting muscle, the actin filaments have their binding sites blocked by a protein complex called tropomyosin, which is held in position by another protein called troponin. When calcium floods into the sarcomere after electrical stimulation, it binds to troponin. This binding causes a conformational change, a shift in the physical shape of the protein, which moves tropomyosin out of the way, exposing the actin binding sites. The myosin heads can now attach. Contraction begins. When the electrical signal stops and calcium is pumped back into the sarcoplasmic reticulum, the binding sites are covered again, the myosin heads detach, and the muscle relaxes. This on-off switch, regulated by calcium and a pair of proteins, is what allows the body to control contraction with extraordinary precision. The system is modular by design. A single motor neuron does not activate just one muscle fiber. It activates a group of them, collectively called a motor unit. The size of a motor unit varies enormously depending on the muscle's function. The muscles that control the fine movements of the eye contain motor units with only a handful of fibers each, allowing for extremely delicate adjustments. The large muscles of the thigh, which produce powerful but less precisely calibrated forces, contain motor units with hundreds or even thousands of fibers. When the brain wants to increase the force a muscle produces, it does so by recruiting more motor units, a principle called motor unit recruitment, and by increasing the rate at which individual motor neurons fire. These two mechanisms together allow for smooth gradations in force across a remarkable range. The muscle fiber itself is not a simple, uniform structure. Fibers are classified into different types based on their metabolic characteristics and the speed at which they contract. What are often called slow-twitch fibers, more precisely type 1 fibers, are dense with mitochondria, rich in oxygen-binding proteins, and highly fatigue-resistant. They are the workhorses of endurance, the fibers that carry you through hours of walking or cycling or standing. Fast-twitch fibers, particularly type 2X, are built for speed and power. They generate force rapidly, but exhaust quickly. Between these two extremes sits an intermediate category capable of some degree of adaptation. This fiber-type distribution is partly genetic and partly the result of training, which is why endurance athletes and sprinters have distinctly different muscular profiles, even when their overall muscle mass is similar. One of the more surprising aspects of muscle physiology is how much the system involves communication beyond simple nerve signals. Muscles produce and release signaling molecules, sometimes called myokines, that travel through the bloodstream and affect organs throughout the body. Exercise-induced signals from muscle tissue have been linked to effects on the brain, the liver, the immune system, and adipose tissue. Evidence suggests that muscles function not merely as mechanical actuators, but as active endocrine organs, participating in a broader conversation about the body's metabolic and physiological state. Research continues to explore the full extent of this signaling network, and there is still no complete consensus on how many myokines exist or what all of them do. There is also a dimension of muscle physiology that touches directly on the limits of human performance. The relationship between force production and velocity, the fundamental trade-off in muscle mechanics, explains why no single muscle fiber can be simultaneously maximally strong and maximally fast. Producing peak force requires slow, controlled contraction. Maximum velocity is achieved only at zero load. This trade-off is not a design flaw. It is a consequence of the underlying biochemistry of myosin cross-bridge cycling. Elite athletes in different disciplines are, in a very real sense, optimizing for different points along this curve. And the body, through training and adaptation, restructures itself at the cellular level to serve those demands. What is perhaps most striking about all of this? The action potentials, the calcium floods, the sliding filaments, the myosin power strokes, the motor unit recruitment, is how completely invisible it all is. You raise your hand, and none of this is apparent. The complexity is buried so deeply in the architecture of the cell that it requires an electron microscope and decades of biochemical research to see it at all. And yet it is happening continuously in every muscle of your body, every moment of your life. The story of how muscles move is, in one sense, the story of how life translates information into action. The signal begins as a thought, or a reflex, or an autonomic command, and ends as force, movement, work performed in the physical world. Along the way, it passes through electrical gradients, chemical messengers, calcium ions, and proteins that change shape in response to molecular signals, driving one set of filaments past another with a precision that no human engineering has ever replicated at that scale. This machinery does not wear out in the way artificial machines do. It repairs itself, adapts to new demands, and maintains its function across decades of continuous use. There is something worth sitting with in that. The body you inhabit is not simply a biological structure that happens to move. It is a system of extraordinary mechanical and chemical complexity, refined over evolutionary time to do exactly what it does with minimal waste and maximal reliability. Every contraction you have ever made, every step, every breath, every grip of a hand, has been produced by the same molecular mechanism. The same sliding filaments, the same calcium switches, the same ancient proteins. And in the quiet architecture of that mechanism, there is something that looks, from a sufficient distance, not entirely unlike design. And the next time you move, even something as simple as lifting your hand, remember that beneath that effortless action lies a microscopic symphony of electrical signals, calcium surges, and molecular machines working with astonishing precision every single second of your life. If this changed the way you see your own body, make sure to subscribe to Anatomy Unlocked. Because here, we don't just show you how the body looks. We reveal how it truly works.